So thank you, Janet, and welcome everybody. Welcome to the fifth event. I was arguing with Maury earlier on whether this was the fourth or the fifth event. It's the fourth in the UK and the fifth in Europe. I think the first one was a very small event in Brussels, I think. And when we talked about holding a bigger event in the UK, I think it was 2014, we were kind of worried if anyone would show up. And a lot of firms were still running on vapors right at the beginning. And it's all changed now. And it's all changed because of some fantastic clinical results. We did a little survey earlier in the year just on immuno-oncology, or immunotherapies rather, and we found 32 firms in the US, 25 in Europe, and 12 in Asia. And now there's been lots of VC input. The public markets are really good, and of course the first major takeout with Kite and Gilead. So it's a really hot area, and I'm really pleased today to have two CEOs of two companies in Europe, a longer established firm with Medica with Gregg, and a more recent one with Eric with PDC Line. So I'm going to briefly introduce both Gregg and Eric, and then ask them to tell a little bit about the companies. Now, Gregg's got a really interesting background. He was an investment banker, and I guess like all CEOs, money's never far from your mind, which is why we're here. But Gregg kind of saw the opportunity in immunotherapies quite a long time ago now.
10 years.
And 10 years ago he saw this, and he went and thought, if I'm going to be in this space, I need to know more about it, and put himself on an MSc to learn the whole thing, which I think was absolutely remarkable. He gathered a team of immunologists together, initially went down CMV, transformed cells route, some good products there, but more recently with CAR-Ts and TCRs. And in fact, licensed from my organization some technology just recently. And Eric, as probably more recently as the CEO of Promthera, he's serial entrepreneur, and recently has helped found PDC Line, which is a spin-out from the French Blood Service. So I'm going to start with Gregg. Really easy one, Gregg. Tell me about Medica, what you've been doing, and where you're going, and then we'll get into more general trends.
Right, okay. Well, thanks, Keith. I won't give you the full 10-year history of Medica. I think I'll start in 2016, when we announced two major collaborations which really set us up in the CAR and the TCR space. So in the T-cell receptor space, we announced a major co-development effort with University College London, Professor Hans Staus, and we exclusively licensed technology that he had, which basically improved the performance of any engineered T-cell receptor by increasing the expression of the T-cell receptor on the surface of the T-cell. So very powerful technology that can apply to any T-cell receptor. And that led us to working together with Catapult, because they had a T-cell receptor under development, the WT1 T-cell receptor, which was really interesting. They were running a clinical trial, had done a great job in setting that up. And so we decided, let's put together this new technology with Catapult's WT1 TCR in the clinic and really take this forward. So we continue to work on that, and we continue to build our pipeline around the TCR space as well. In the CAR space, again, in the same year, 2016, we announced a major broad-reaching collaboration with Baylor College of Medicine in Houston, Texas. And they're one of the real leaders in the development of CAR technology. And we actually have 4 separate products under development with Baylor College of Medicine. And I'm very pleased to say that we just filed our first IND last week. It's not announced yet. So we will start treating patients very shortly with our first CAR product in the US. So in a very short period of time, we'll be in the clinic with an engineered T-cell receptor and our first CAR product, which will place Medica in a very small group of companies to have both technologies at the clinical stage. So we're moving ahead.
Good. So, and Eric, I mean, your technology is in the cancer vaccine space, so very different. And I think one of the good things about today is hopefully, everyone thinks immunotherapies are, all CAR T, TCRs, but of course there's a whole world of other therapies out there as well, from the cancer vaccines through NK, Tregs, gamma delta, macrophages, all kind of bubbling away in the background. So tell us where PD lines come from.
Okay, so PDC line, as you said, is a spin-off from the French Blood Bank. In fact, the scientific founder has identified in the blood of leukemia patients specific cells that have the ability to grow in culture and very high potency in terms of expansions of what we call the T cells, which are that kill the cancer. And 15 years ago, and based on this work, he developed a stabilized cell line that grow now in culture without any calf serum in a synthetic media. And first clinical trial in melanoma has been accomplished in 9 patients with interesting results. The company has been founded in 2014 based on that, and I joined the company beginning of 2016 to develop further the organizations. So you're right, the vaccine area and immuno-oncology in general is a booming area. So if you come back 10 years ago, nobody would believe that. It has been very difficult because the first generation of dendritic, for example, has been an issue, and even the product that has been put on the market, the first one, we cannot say has been a big commercial success. But now with the new generations of products, the launch of the checkpoint inhibitor, I think that it has been one of the first corner, checkpoint inhibitor that destroy the defense of the tumor, that opened the door to a lot of new combinations. And vaccine in combinations with this checkpoint inhibitor, we believe would be strongly more potent and more with clinical benefit that we have seen so far. So I think it's an exciting area for immuno-oncology and definitely for vaccine. Today a new hot topic is, when I say hot, it's because there is a lot of investment ongoing, is on neoantigens and personalized vaccines. Right. So there is potential on it, but as well challenges related to the logistic and the commercials of these neoantigens. But it's a field to follow, and we are following as well this area because we'll have a program in development in this area. So shared antigens and neoantigens, which are the mutated antigens coming from the tumor, specific tumor of the patients. It's really personalized.
So it kind of reminds me a little bit, the kind of cancer vaccine area is starting to get hot again now. And it reminds me a little bit of gene therapy in a way, which was hot, 10 years or more ago, really bombed out the scene, and then came back really, really strongly. And of course, cancer vaccines were hot, kind of bombed out the scene, and now coming back strongly. And of course, for the gene therapy side, the clinical academics went back and fixed the kind of things that were wrong with the technology. Have you got a similar kind of view of how those technologies were evolved in that space?
Yeah, I think science needs to evolve and maturate. For technology need to maturate. We have seen that in monoclonal antibodies as well. A long time ago, you had the fully mouse, the chimeric, and the fully human. So I think it's a normal system with maturations. And in therapeutic vaccine, once again, the checkpoint inhibitor will open the door, because it was extremely difficult to have significant clinical benefit with this defense of the tumor, that deactivates the T lymphocyte. Mm-hmm. The next generations of vaccine, and we believe we are in this area, with a lot of potency off the shelf. We are off the shelf, so you resolve the issue of logistic, of taking the blood of the patient and making the dendritic from the personalized patients. You have new products with vaccine based on bacteria, vaccine based on viruses, And I've seen that just 2 days ago, a company like Nuscom raised 42 million euros as a A round, I believe, or B round. It was really the beginning, and they are not yet in clinical trials. So just to show you the potentials. And company neuroantigen like Neuron, not Neuron, it's a US company or a German company based in Viscera raised more than first round of financing 50 million euros. So you see the potential that at least the financial community see in Viscera. It's BioNTech, for example. So it's moving, and I think in the right directions.
Well, we'll kind of come on to some of the kind of, you mentioned manufacturing, we'll come on to that in a little while, but I'm going to turn back to Gregg now and really get your perspective, Gregg, on, after the initial fantastic results, there's been an explosion in kind of more and more segmenting of the technology and second and third generations. Where do you think those kind of trends are taking the CAR-T and TCR space?
Well, I think in both cases, it's all about dialing up the potency of these cell therapies to be able to address solid tumors, because that's the big objective at this point in time. So the CD19 CAR-Ts, absolutely fabulous results in ALL and CLL. But so far, the results in solid tumors for CAR technology has been pretty limited. So the question is why, and what do you do about it? I think we all understand CD19 is a very special target, and T cells and B cells communicate in a very specific way, which is at the heart of the CD19 CAR technology. But I think the real focus at this point in terms of the next generation of CAR and TCR technology is about managing the immunosuppressive tumor microenvironment around these solid tumors. I think we all understand, if you've looked at this field, how important that is. And there are different concepts out there now, and this will really determine who's going to be the leader in this huge space. So our approach is basically relying on two technologies, and this is in partnership with Baylor College of Medicine. So we use IL-15 as an activating cytokine, and when you engineer the CAR into the immune cell, into the T cell, you then also engineer the secretion of IL-15, which is a very important activating cytokine for T cells and also is associated with the memory phenotype, so a long-lived type of cell. And we believe, and others too, certainly Memorial Sloan Kettering is famous for advocating the armored CAR using IL-12, which is a similar concept. So our approach is IL-15, and we hope that that's going to make a big difference in keeping these cells alive and active in the tumor microenvironment. And we'll see patients treated in a very short period of time. So our first indication is neuroblastoma, which is a very difficult solid tumor in children, a very important unmet need, and we'll start seeing the clinical data in just a few months on that.
Great. So you mentioned CD19. I was looking at the kind of trial database the other day in preparation for this, saw a lot of CD33 as well. Mm-hmm. That seemed to be the next, certainly in the blood cancers, that seemed to be a pretty hot antigen.
Yeah, I think there's a number of interesting antigens that are B-cell orientated, and certainly they're being pursued. There's quite a few studies ongoing. And I think that's a very important fertile pathway that people need to pursue because, as I said, these are not necessarily cancer antigens. They can be B-cell antigens. Sure. And so it's using a very special approach that's relevant there. We use classic tumor-associated antigens in our products. So our first target is GD2, which is, I think, ranked number 12 in terms of cancer antigens by the National Cancer Institute. And this is a cancer, oncofetal cancer, antigen where you've got high expression on the cancer and some expression on healthy tissue, but you're looking for that big differential. Right. So for solid tumors, it's a different strategy, really, in terms of high-value targets relative to the blood cancers.
Right. Okay. So Eric, your view of the cancer vaccine space, the kind of where it's going, but also in the kind of NK, Tregs, gamma delta. Do you have in the runners and riders, do you have any favourites in there that you've got in your mind for where PDC line might go next?
In fact, I think the future of the treatment of cancer is by mixing different modalities.
Yeah.
It's one modality for everybody. I don't believe will exist ever in cancer anyway. So new modalities that are in development, even combining CAR-T with vaccine or whatever, I think have a place. The issue now is to identify in these puzzles where to position each technology and to have in details what are the right positioning for each of these modalities. So I think vaccine, and if you ask me a preference, I would say vaccine because I'm in this area, has a place definitely to boost the system, and people are saying it's more in an earlier phase. I'm not completely sure about that, but it's where some of the vaccine are positioning in the pathway of the treatment of cancer. NK, gamma delta are new approach that are quite exciting, definitely. We need to see the clinical result because it's still ongoing. We have already, for the CAR-T cells product that has been approved, but we still have relapse afterwards. So, the answer is not completely there, so there's still a need for new treatments. And I think it's to have a really long-term and to cure or to have a cancer become a chronic disease, you will have to mix modalities.
Yeah.
And maybe mix different modalities according to the time.
Yeah. So I mean, the reason there's all the investor interest and there's the reason the interest, globally in these products is because the results are outstanding, and we've come to expect outstanding results, and there'll be disappointments as well. But one of the things I'm hearing more about is running the combination therapies. How you might do these things, combinations with checkpoint inhibitors. Trialing those is going to be complex. Have you kind of worked out a strategy for that?
In fact, for us, the first logical step is with checkpoint, the checkpoint that are approved in the indication we are targeting. The next one will be in lung cancer, non-small cell lung cancer. The biggest issue today is to get access to the checkpoint because it's a quite expensive therapy. It is in the standard of care, but even though it's not easy to get it paid, by the system or the clinics, the hospitals, and you have to negotiate case by case or to try to have a deal with BMS, Merck, and the others. So which is not easy. So I would say today is one of the biggest hurdles for vaccine therapy that want to develop combinations, and there's a lot of combinations. I've seen that more than 300 clinical trials are ongoing with checkpoint inhibitor. So it's difficult to get patients, it's difficult to get access to patients, it's difficult to get access to checkpoint. So I think it's today the main issues. But the first logic step, from my point of view, is checkpoints. And after, you can, based on good clinical data, you can think about over combinations.
Yeah. It's interesting that you talked about access to patients, and I think I read some information in the UK that several years ago there was only a small percentage of people on a clinical trial in the UK. Now something like 50% of all cancer patients are on a trial. So, with the explosion in the explosion in therapies, how do you convince the clinicians to try your trial versus somebody else's trial? Gregg?
This is actually a big challenge, and there are so many studies going on at this point in time that every cancer patient, is a rare asset at this point in time in terms of developing these products. So we have to fight hard to get the patients into our studies. Of course, the patients are very clued up. Yeah. They're reading the internet, they're talking to their clinicians. They're avoiding, for example, in phase 1, phase 2 studies, they avoid the first cohort where the dose is low because they want a proper dose. Yeah. It's amazing how they're analyzing the clinical trial system. And so it's interesting because you have to market your treatment to clinicians in a very proactive way as you launch a phase 1 study. And you have to really convince the clinicians that your technology is going to make a huge difference and really separate it from all the other studies that are going on. And you also need the help of the advocacy groups and the patient groups. So neuroblastoma is a pediatric condition, and there's an organization called the Alex Lemonade Stand Foundation in America, and they've actually agreed to fund part of this study, and that's hugely important because they have tremendous pull with the parents of these children. So you do have to think very creatively about how you get your patients into your study at this point in time.
Yeah, and it's all well and good going from where the original studies were with the original clinicians, where they had total control of their environment, to, you know, these big multi-center studies. It's tough. Have you experienced that?
Yeah, of course. It's enrollment of patients is definitely an issue, but the only way to handle that is to prepare, you know, to have the right resources internally, to have a good CMO, good staff, which is as well not easy in immuno-oncology because the demand is extremely high. Secondly, it's to have, of course, you convince the clinicians by science, you know, so you need to demonstrate with your preclinical package, and if you have already clinical data, why you are good, why the patients would benefit your products, and make it in the right way, you know. And so I think it's a mix of preparations, a mix of developing a relationship, and proximity with the different centers and clinical centers that make the difference. But there is no, I think it's no rocket science. You just have to go to the field and defend your defend your technology.
Okay, so I'm going to move on to manufacturing now, because you know, you've got to make these things, you've got to make them effectively, you've got to get them to patients, you've got to do all sorts of things which are just taken for granted in small molecules and largely in biologicals these days. So, Gregg, you know, these therapies were handcrafted relatively recently, and the industrialization process and the challenges in there, which is, you know, in all of the things that you've got to think about, is that one of the things that keeps you awake at night?
Well, I think manufacturing is absolutely critical to the success of any cell and gene therapy company. And we're just in the first chapter of understanding how to manufacture these products. The companies that get it right will own the space, and they'll end up licensing in all the technology and the products from the companies that don't get it right, in effect. So manufacturing has always been important to us, deeply important to us, and we've been manufacturing products since 2008, so we have a fair amount of experience. We've gone through several models, and we continue to evolve the model that works best for us in our cell therapies. But I think the key point for us at this moment is that the CAR-T manufacturing technology, the TCR manufacturing technology, still needs intensive process development work before we can really scale up and manufacture these in the tens of thousands, you know, for the solid tumor applications. So our emphasis is not so much on can we, you know, buy a big swanky manufacturing facility, but actually, what is the science that we're using to manufacture the product? And so we look for very efficient infrastructure while we conduct the process development. So we're very happy that we were able to join you up in Stevenage, and that was a great solution for us. You built the infrastructure, and now we're in that facility, and we're focusing on our particular products.
I was hoping you were going to say this. It wasn't. Good advert, guys.
Yeah, no, but it works very nicely because we can invest in where we create value at this point in time, and we don't have to worry about the infrastructure. And it's a great benefit for us. There's no doubt.
Great. And Eric, I mean, your technology came out of the French Blood Service?
Yeah. In fact, so the issue of manufacturing is is the third cell therapy company I'm involved in, Myosix, Prometera, and now PDC Line. So manufacturing has always been the key issue. So I think in PDC Line, what we try to do is based on all the experience I've tried to accumulate and the scientist has made, is to simplify as much as we can the process, try to get rid about, to work on a synthetic media, try to work on, we have cell in suspension, so it simplify a lot, you know, because a lot of equipment, bioreactor are already existing because they have been developed for monoclonal antibodies for the cells in suspensions, and to be able to streamline the process. But we benefit as well to the big expertise of the French Blood Bank that had GMP facility in-house, and they collect the blood, which is extremely regulated in France, and they have a very strong know-how in terms of manufacturing quality assurance, and they provide a lot as well in terms of improving the process.
Great. Well, this is an investor conference, so I'm going to finish on where next. We've got a couple of minutes to go, so really brief. You're early in the life of the company, and you're looking to raise an early round. And of course, Gregg, you've had several rounds. So, in 1 minute, what are you looking for, and who do you want as investors?
Okay, so we are raising. I'm raising for a second round of financing. We are looking for 24 million euros, including the grant and subsidy of the Western regions that we get already. So it's 18 million in equities. We have already a big part of it. But we're still looking for new investors to close the round, because we not make it as long as we don't have the full money. And investors that have interest in this area and is definitely welcome to discuss with me. So this is corporate venture. I like the mix of corporate and classical VCs. Based on previous experience, it works quite well.
Okay, and Gregg, your thoughts about where your investment needs are? Obviously, you had a decent raise this summer, and of course, it's never enough. So
it's never enough, that's for sure. Yeah. So we've raised $150 million, roughly 130 million pounds, over the past 10 years, actually, but most of that in the past 3 to 4 years. We raised 60 million pounds, as you mentioned, earlier this year. And that will take us to kind of mid-2019. As I mentioned, we're going to be seeing clinical data beginning in the next few months.
Right.
So we think by the time sort of late 2018, early 2019 rolls around, we'll have sufficient clinical data to start thinking about our next financing, which for us most likely, hopefully, will be a crossover round in the US, where we will lay the groundwork to do a NASDAQ IPO in the mid to later part of 2019. That's our plan if it all goes well.
Okay, great. So I think I've burnt all the time. So there's no time for questions. I'm sure if anyone's got questions, form an orderly queue with checkbooks. And thank you very much to Gregg and Eric.
Yeah.
Thank you.